Hello, we will get started as soon as Zoom lets everyone into the webinar. Just a few seconds or so. Okay, well, welcome everyone. This is Christy Sullivan, and I'm the Secretary of the American Society for Cellular and Computational Toxicology. Our May webinar is, as always, presented by ASCCT and the European Society for Toxicology in Vitro. I want to encourage you to visit both of our websites to find out more about our organizations, support our activities by becoming a member. That will help us continue to put on programs like this webinar, other activities, and annual meetings. So the webinar is recorded, and the recording will be posted on the ASCCT website a few days from now. In the webinar platform, you can ask questions of the presenter using the Q&A feature at the bottom of your screen. There's a toolbar with that Q&A feature. Please do use the Q&A, not the chat, for your questions, because it's hard to keep track of questions in two places. You can put in your questions at any time. We will have a Q&A after each speaker. So as soon as, while the first speaker is speaking, if you think of a question, pop it in, and that way we can get rolling. We have also enabled closed captions. You can turn them on or off if you like, according to your preference. And finally, please say hello in the chat and let us know where you're joining from. We always like to see who's with us. All right, so a couple of announcements. First of all, ASCCT is having its annual meeting in October of this year at the Institute for In Vitro Sciences, my organization in Gaithersburg, Maryland. I do see that the deadline for proposals is passed. In fact, there are still a couple of spots open for sessions, so get in touch with us if you have an awesome session idea that you want to propose. We're going to be opening up abstract submission probably in the next couple of weeks. And then let me also now turn it over to ESTIV Vice President Clive Roper to tell you a little bit about what's going on at the Society.
Hi, yeah. Thank you very much, Christy. Yes, the ESTIV, you're going to have to wait a little bit longer for our next meeting, as we meet every 2 years. Our next meeting, as you can see here, will be in the summer in Maastricht in the Netherlands. Just to give you a little bit of update on where we are there, the proposals for sessions have all been actually now approved, and we've provisionally approved those proposals, so they will appear on the website shortly. And but there's plenty time to get your abstracts in. So the abstracts module will be opening on the 27th of May. I think that's great. And of course, you want to learn more, go to the website, become a member. Two great organizations together, ESTIV and ASCCT. And we also shouldn't forget our Japanese colleagues, the JSAAE. And that brings me nicely into to get towards the first speaker. I was very lucky to go to all 3 conferences last year, and we were able to give an ESTIV award in both Japan and at ASCCT. So our first presenter here is going to be Randolph from NeurosetteArray, talking about the RosetteArray platform, AI-enabled high-throughput screening of human neurodevelopmental for toxicology and precision medicine. And over to Ashton.
Well, let me go ahead and read Ashton's bio just to give a little bit more info there.
Sorry.
No worries. So Dr. Ashton received his bachelor's from Hampton University and PhD from Rensselaer Polytechnic Institute in chemical engineering. And during graduate studies, he researched how engineering biomaterials at the nanoscale could regulate the fate of adult neural stem cells. He continued to pursue his interest in stem cells and tissue engineering, as a California Institute for Regenerative Medicine and NIH postdoctoral fellow at the University of California, Berkeley. In 2011, he joined the faculty in Wisconsin Institute for Discovery at the University of Wisconsin-Madison, and in 2018 was promoted to Associate Professor of Biomedical Engineering, as well as Associate Director of the UW-Madison Stem Cell Regenerative Medicine Center. The goal of his research is to engineering human tissues that can be used as tools or therapeutics to prevent or cure central nervous system disorders. His lab currently melds state-of-the-art biomaterials approaches with novel human neural stem cells derivation protocols to bioengineer brain and spinal cord cells and tissue models in vitro. And then finally, in 2021, Professor Ashton and collaborators embarked on a technology startup, NeuroRosetta LLC, to translate the lab's innovations for developmental neurotoxicology, neurodevelopmental disease modeling, and drug discovery applications. The reason that both of these presenters are presenting today and that we have Clive joining us as well is because we have these presentations won awards at our annual meeting. And so Clive joined us to provide Randolph in last October with an ESTIV award. And Laura, the second presenter, won an award from ASCCT for her presentation. So that's why they're presenting their work to you today. Why don't we, I will introduce Laura once it's her turn to present. So we'll start with Randolph. Let me just tell you also to go ahead and go to the website to register for our next webinar, which is going to be June 10th. And that's going to be Adam Wood from Unilever and Next Generation Risk Assessment for Occupational Safety. Okay, so I will stop sharing, Randolph. That will be your cue to go ahead and take the floor.
Okay, can everyone see my screen?
Yes.
Great. So thank you for that kind introduction. My name is Randolph Ashton, Associate Professor of Biomedical Engineering at the University of Wisconsin-Madison. And of course, with this presentation, I have a huge conflict of interest, which is that we've tried to translate this so this can be accessible to everyone, technology that we developed in my lab here at Wisconsin via our biotech startup, which we call NeuroZetta, which hopefully will become apparent as to why that is the name. And I want to acknowledge at the outset, we had a STTR grant from NIEHS, and that was really how we were able to get this company off the ground. And today I'll talk to you about our RosetteArray platform that we broadly use for quantitative high-throughput screening of human neurodevelopmental risk with applications in toxicology and precision medicine, which I will talk about here as well. So current technology that's available to companies as recommended by the DNT in vitro battery from the OECD guideline recommendation consists of about 17 different 2D cell culture assays, 2D and 3D cell culture assays that allow you to look at and model different stages of human brain development, largely brain development. And this is a nice tool. It includes about 17 different assays that some are commercial and can be used, and I'm not sure if the entire platform is commercial, but at least a good portion of these are commercially available. Now, one thing that, as you can see from this schematic, is there are not as many assays on sort of the 0 to 6 week stages of CNS development, and this is a critical time point as the pathogenesis of birth defects such as neural tube defects, which are detected within the second trimester of pregnancy. Autism spectrum disorders, this is a type of neurodevelopmental disorder, and there are other neurodevelopmental disorders that can be detected within 24 to 48 months after birth. And even predisposing people to Huntington's neurodegenerative disorders such as Huntington's disease, the pathogenesis for those disorders is in part thought to occur very early in the formation of the human central nervous system. And this is a critical stage to test and be able to model because it is prior to having some of the innate barriers that form that could potentially protect the CNS from chemical exposures. And so a technology that is now sort of state of art in the laboratory sphere for us neuroscientists is the use of these neuroorganoid models. And so these are 3D organoid models generated from human pluripotent stem cells where you culture them in a 3-dimensional structure. and they can start to recapitulate some of the morphogenesis aspects. That's both differentiation and formation of tissue cytoarchitectures that mirror what happens in neurodevelopment. So this is a very powerful tool, has really transformed how we approach modeling central nervous system development and disease in the laboratory. And when we started developing the technologies that I'm going to present today, this really had an issue with reproducibility and scalability. And still, I would say reproducibility of tissue structure, as well as scalability of the platform in general for something that can be used at a clinical level is still a challenge unless you have a lot of money. And so what our RosetteArray platform does is it sort of hits this window where there's not a lot of good representation from the DNT IVB and the stages going from the human pluripotent stem cell to the initial formation of the earliest neural tube-like structure, which is the analog for all central nervous system tissues. And so we're modeling this in both forebrain and spinal cord RosetteArrays, and we have translated this into a scalable platform. So what we're doing is essentially modeling the formation of this early neural tube structure from which all brain and spinal cord is generated within the human central nervous system. And if you take a transverse slice of this at any point along that head to tail axis, you can see that at the beginning of this neural tube structure, it is essentially a single layer of these neural stem cells where the cell bodies are stained here in blue, and they have one foot of those cells on the apical portion. This is the portion that will line the initial neural tube canal, which will become your ventricles and your spinal canal eventually. And then they have another foot on the basal portion where they deposit extracellular matrix proteins, and really that walls off the CNS very early in development. And what we've known for the past 25 years is that if you differentiate human pluripotent stem cells to these neural tube-like neural stem cells, then they spontaneously try to recapitulate or form this type of neural tube structure, which in standard 2D culture, as well as some 3D cultures, we call this a rosette. And so this rosette, each one of these rosettes is a center of morphogenesis and is capable of generating various neuronal and glial subtypes that are associated with that particular level to which of the neural tube to which the CNS is patterned. And so this is a very powerful tool, but as you can see in standard 2D culture and 3D culture, this is a spontaneous process. And so the generation of multiple rosettes of various shapes and sizes causes randomized neural organoid structure under standard protocols. And this can lead to the necessity to have protracted derivation protocols to generate the neuronal and glial cell types that come after that. And then you're analyzing a tissue that is typically 0.5 to 1 millimeter in size. So you have to section it or do some fancy clearing in order to analyze it. And so the process for generating and analyzing these tissues in mass is very cost and time prohibitive when you try to do it at scale. And so what we've done in my lab is we've demonstrated that you can integrate micropattern culture substrates in order to provide an optimal shape for your initial cell aggregate. And these are adherent cell aggregates. And if you let them grow and start to morph, then they can preferentially generate this single polarized rosette structure, better mimicking the single neural tube-like structure that forms within the human embryo. And this gives you a rapid, reproducible, and scalable platform in order to derive thousands of these across a plate. And you can do that, of course, in parallel to run screening assays. What we've done at the startup is also integrated the platform with image analysis, which we call RosetteArray platform software that is now available online. And so you can take automated confocal microscopy Z-stacks of the tissues across the plate and then run them through this software, and it would detect various features of this tissue, including the polarized rosette structure. So in the time that it would take you to analyze around 9,000 different neural organoids with normal protocols, that would be really over 6 months. We can now do this at the rosette stage within about 2.5 weeks for the same number of tissues. And so this really gives us a nice quantitative screenable platform to try to use for various applications. And while the tissues that I've shown you so far, I'm showing you confocal slices, so that would be a two-dimensional plane within a larger tissue. I just wanted to show you that when you see the pluripotent stem cells on the micropatterned arrays, they initially start off as a monolayer, but they proliferate, of course, and undergo morphogenesis such that you get a three-dimensional structure where that polarized rosette is in the center of that. And if you just let them continue to grow, then they'll form three-dimensional neuroepithelial organoids that just sort of bud off and can give rise to full-blown cortical neuroorganoids, or whichever organoid for the region along the head to tail axis you're patterning your cells to. And so the platform that we run here at UW-Madison at Neurosetta, as well as what other people have started to run in their labs, is that they can essentially generate pluripotent stem cells from various control or disease patients. And they freeze them down as cell banks that they can then thaw and seed directly into these micropattern well plates, which we sell commercially at Neurosetta. And over the course of a week, they can watch the tissues grow, expose them to various chemicals or drugs. And at the end of that week time period, they'll have an array of around 9,000 different nascent neuroorganoids across the plate. And they can go through and image that with automated microscopy and then run it through our software in order to detect whether you have proper tissue formation. And so this really gives us an unprecedented quantitative high-throughput screening of nascent neuroorganoid culture, which of course in my laboratory, we're extending further. But as it stands, this RosetteArray platform we think is pretty applicable for developmental neurotoxicity testing of various chemicals and drugs, as well as for modeling various neurological disorders, such as birth defects, neurodevelopmental disorders, and even predisposition for neurodegenerative disorders. And what we've also done at Neurosetta is worked with the EPA in order to integrate our results such that you can get outputs in the TCPL Fit2 format. And we're finalizing this model right now from a chemical library screen. But the readouts that we are providing currently are the number of tissues per well as sort of a general measure of cytotoxicity. And we're also looking at the presence of the proper differentiation into these neural tube, neural stem cells, the number of those neural tube, neural stem cells that you have, and then of course the formation and the regularity of that rosette structure. And so what our platform, we believe, allows now is for standard discovery and development pipelines where you really cannot test on human developmental neurotoxicity. Now you can do this at a stage upstream in your discovery process where you can really, for 10 to 100 compounds, you could use this RosetteArray platform to screen for things that cause developmental neurotoxicity hazard. And of course, that can save you tons of money in the bottom line by not proceeding forward with chemicals that would eventually have potentially some detection of human developmental neurotoxicity, ultimately lowering the risk, time, and cost for this entire process. So as a fundamental question, of course, you want to ask how good is our RosetteArray assay at predicting human developmental neurotoxic hazard. And so what we started with first is just showing that we could screen chemicals that are solvated in dimethyl sulfoxide, sort of a common solvent for chemical libraries. And what we see for both our forebrain assay on the top, when you're looking at the number of cells per tissue, the number of PAX6 positive neural stem cells per tissue, and the number of tissues that give rise to that single rosette structure, you really don't have any inhibition of our process until you get upwards of 0.1% DMSO. And so you can basically now do a 1 to 1,000-fold dilution and not have any significant effects in our assay for chemical solvated in DMSO for both the forebrain and the spinal RosetteArray assay. This is a pattern to the lumbar spinal cord. We looked at reproducibility, and for our forebrain assay, we did this ad nauseum. And we see that across multiple plates, whether you're using a semi-automated liquid handler to seed your cells and change media, or you're using a multi-channel pipette manually, the number you see here in the plate, we use the interior 60 wells. Across those 60 wells, this is the efficiency of single rosette formation that we were able to generate. And this has held across multiple assays, and you can see sort of the homogeneity of that across different wells. Of course, there's a little bit of variability there, but across most wells, it's pretty homogeneous in terms of the uniformity that you can generate, and across assays. We've done this now for lots of different assays, and this number is actually ticked up to 0.56 now for our Z-factor score. But on every plate, we have a solvent control, so that's no chemicals, just the cell culture media. And then we also have methotrexate, which is a compound that's well known to cause a neural tube defect risk or to disrupt formation of that polarized rosette structure. And so looking at the comparison between the efficiencies we get or the knockdown we get with the positive control, we have a Z-factor here of 0.532. And again, we've done this now for over 60 assays, and it's about 0.56. We can also integrate simulated human metabolism into our assay. So this is with just a S9 pooled human liver fraction pre-digestion. We've shown that those components can be tolerated with our media, where we can do the pre-digestion at the 2 mg per mil level and then do a 1 to 1,000-fold dilution or lower in order to create the dose response curve. And so you can have that initial pre-digestion and then test your molecules in the assay. And we really don't see an effect until you go above that 0.002 mg per mil level on our 4-brain RosetteArray assay. We haven't tested it just on the spinal. And for our 4-brain RosetteArray assay, some of the standard results that we've gotten when looking at compounds that have been tested in the DNT-IVB, you have compounds where you should see really no hazard associated with them, such as sugar or aspirin. And you have other compounds where you can see some hazard, such as permethrin, but that's at pretty high levels. The dotted lines you see here are assays in the DNT-IVB, and they are the IC50s for those assays as well. And then we have things like imectin benzoate, where you have a medium risk or medium hazard, as we call it. And you can see the IC50 is creeping towards the left. And then you have things that have high risk, things that are known to cause, for example, neurotube defects or neurodevelopmental disorders if they're exposed to patients. And those tend to be pretty easily detected by our assay, particularly within that single rosette emergence metric, which is the green line you see here. And we've embarked upon a chemical library screen. And so we're doing this in collaboration with the EPA, and this is funded by the National Institute for Environmental Health Sciences, where we're basically doing 8-point dose response for chemicals that are also in that OECD IVB battery that have been published. We're not doing any metabolism here, just a standard 4-brain RosetteArray screen as we try to push this forward in the regulatory process. So this is a blinded screen that we're doing with the EPA, who provided the compounds to NeuroRosetta. And again, funded by NIEHS. These are the results from our first 50 compounds. We're going up to 90, and we are just about finishing up. We're doing our last panel of compounds now. So hopefully we'll have this out for you in the fall, at least as a preprint, if not a accepted paper. And so what we see so far with these first 50 is we have pretty good sensitivity and specificity. Of course, specificity is limited because of the number of compounds that we've tested. We're trying to push this up to at least 10 more compounds with the second set of 40 compounds. So we'll have some updates to those numbers. But what we can tell here is that this is hitting it in line with sort of the sensitivity and specificity of the DNT-IVB. And so we don't have the final numbers yet, but we hopefully will have those soon in order to do the direct comparison. So overall, we think our assay, at least the 4-brain RosetteArray assay, is pretty predictive of human developmental neurotoxicity hazard. And we are pushing forward with that in the regulatory process soon as we complete our library screen. And so the second question that we ask, because as you heard from my background, I'm actually a chemical engineer or a stem cell bioengineer, as some might say, versus a toxicologist. And so we started off pretty early in the process was trying to see if we could predict whether the assay is good for predicting human neurodevelopmental risk overall. So things that would affect human neurodevelopment and actually have clinical correlations with that effect. And so that's something that I will talk about now. And really the question we were asking is, does our model predict things that would take a pregnancy from the birth of a baby with normal cognitive and motor function and lead to a pregnancy that is affected by a congenital birth defect, such as spinal bifida, or a neurodevelopmental disorder, such as autism, or a predisposition of that baby to have a neurodegenerative disorder, such as Huntington's disease. And so we've had publications or manuscripts now in all three of these. Two at the top, these are ones that we've put out. One that was a Nature Neuroscience publication where we modeled the risk for autism spectrum disorder, a genetic risk model. And then we also have our paper that's in preprint now, hopefully out soon, where we have modeled chemical and genetic factors that can cause spina bifida risk. Another paper has been put out by a different group, which they show that the Rosette assay can be used for predicting predisposition for Huntington's disease. And the real question that we're asking here, that everybody's kind of asking, is how does a person's genetics, which can include not just single mutations, but often polygenic etiologies with their disorders, interact with the environment that they come into contact with, the thousands of chemicals that we have in our everyday lives, and can those cause a risk for certain patient populations based on their genetic background. And so this question is something that we really think our RosetteArray assay is really primed to be used as a screening platform to address these questions. And so that's what we're trying to push forward. So starting with neurotube defects, particularly looking at chemical factors that are known to cause those risks, we tested those in our RosetteArray assay. And so this is very early on. So we're only showing the 3 different metrics here because we hadn't started to generate that tissue per well metric. But when you look at things like glycolic acid, you see no effect. This is something that's naturally found in the body. But when you switch over to methotrexate, again, the positive control that we use, one that's well known to cause a neurotube defect risk, as well as valproic acid, now you see increased sensitivity of the single rosette emergence metric. In that assay, we get nice dose responses and IC50s. And when you look at things like pesticides that have been pulled off the market because of their association with neurotube defect risk, such as Benamil, here we see a different profile where there's no statistical difference between our rosette emergence matrix metric versus the cells per tissue or the neural induction or percent PAC6 metric. Largely indicating that this Benamil is affecting fundamental sort of cell physiology processes versus potentially methotrexate and valproic acid, which hit the rosette emergence metric about an order or 2 orders of magnitude before they hit the other 2 cell viability and neural differentiation metrics. We also looked at a more contemporary risk factor, dolutegravir, which is used at the 3 to 10 micromolar serum level therapeutic range, so that's pretty high. And in population studies, it was shown that this has about a 3-fold increase in neurotube defect risk. And this has been sustained over multiple studies. And so we exposed our RosetteArrays to a dose response of dolutegravir, and we saw a nice IC50. Largely, these curves overlap, indicating that we're hitting some potentially fundamental process of cell physiology. We then have done basically what they did in other studies, which was looking at folic acid supplementation to show that if you supplement this therapy with folic acid, you can reduce that risk. And indeed, that's what we see, where we can start to recover our single rosette emergence knockdown as we increase the level of 5-methyl tetrahydrofolic acid that's in our assay. And then we looked at both things that are known genetic factors as well as genetic factors in the presence of chemicals known to cause neurotube defect risk. And so when you look at the folic acid pathway, if you hit it with methotrexate, what you're doing is inhibiting upstream this DHFR enzyme in the folic acid metabolism pathway. And what we've shown is that you can start to rescue that if you add in the 5-MT-HFA, which is a downstream production of this initial folate metabolism. What also we know from clinical and epidemiological studies is that a significant portion of the population has a point mutation in their MTHFR enzyme, which decreases the efficacy of that enzyme. And so across 19 different clinical trials, over 6,000 patients, we know that if you're heterozygous versus homozygous for this particular mutation, then the odds of having a pregnancy affected by spina bifida goes up about 40%. And so we created human embryonic stem cell lines, gene edited them with this mutation. Turns out that the H9 line is actually heterozygous for this mutation, as is 30 to 70% of the people listening to this talk. And so we made 2 homozygous lines for that mutation. And we're looking to see if we could see an increase in risk going from the heterozygous to the homozygous phenotype. And so when we ran our RosetteArray assay, it's important to note that all 3 of these lines could make efficient single rosette emergence. So we saw no difference under control conditions. But when we challenged them by adding in methotrexate, sort of further hitting this pathway, then we saw a significant difference between the homozygous lines and the heterozygous line. And all of this could be rescued by adding in the 5-MTHFA supplement, the folic acid supplementation. And so we went back and did a full dose response with the wild type heterozygous versus the homozygous line. And we saw a sustained sensitivity to methotrexate in the homozygous line versus the heterozygous line. And it's important to note that this was done across 2 separate 4-brain RosetteArray plates to show that this is indeed reproducible. And we analyzed over 6,000 of these nascent neuroorganoids or rosette tissues in this process. And it's interesting that when you look at the ratio of those IC50s, you're seeing about a 40% increase in sensitivity to methotrexate compared to the odds ratio that you see with the heterozygous versus homozygous phenotype. And so here we seem to be modeling what's been seen across multiple clinical trial studies in the human genetic background. And so we then also collaborated with a group out at UCSD, the lab of Dr. Georgia Quadrado, in order to see if we can model an autism spectrum disorder risk factor, one that's caused by a genetic background. And so the lines that Dr. Quadrado had, she had a patient that had a SYNGAP1 mutation. SYNGAP1 is a top genetic risk factor for autism spectrum disorder, which we currently believe represents 1 to 2% of all intellectual disability cases. And this patient had a haploinsufficiency in their SYNGAP1 gene. And Dr. Quadrado, she created a corrected isogenic control. And when they ran their cortical organoid protocols, this is a 2-month protocol for them, they generated these organoids. Both of these lines can generate rosettes, but you can see by the schematic some differences in the characterization of the rosettes that are generated, both the number and sort of the organization of those SOX2 positive neural stem cell containing rosette zones. And they noted that by analysis, sort of painful analysis of their organoids. And so I gave this presentation, not with this particular example, but and Dr. Quadrado said we think we might be able to use that to better quantify our rosette phenotype that we seem to see in our autism lines. And so they ran the patient line here. As you can see, we sent them the plates and they ran this on their own, as multiple labs are doing now. And they were able to generate these tissues really after their second try. As long as you have somebody who's familiar with pluripotent stem cell culture, you basically seed it and run it like a standard 96-well plate. And they saw limited rosette formation in their patient line. And then when they ran their corrected isogenic control, they saw a significant increase in the number of single rosettes that emerged in that control line. And I just want to bring up again, this was a 8-day assay versus their 2-month cortical derivation protocol. They then actually, in order to show that this was reproducible for that particular genetic mutation, they took the iPSC control line and engineered back in that mutation into that iPSC control line. And here they saw even better results in that iPSC control line's background for the inhibition of single rosette formation. So I just want to note it's important that what we've done here is standardize the formation of these single rosette tissues within space and time. So it's not that a line can't generate rosettes, it's just that something about its intracellular signaling pathways via the genetic background that's in that line is causing a perturbation to that process that you can easily pick up because we've standardized the formation of these single rosette structures. And that goes for chemical exposures as well. And so what I've shown you today is that our RosetteArray platform, we can do this for 4-brain and spinal. Most of the results I showed you today were 4-brain. It works with chemicals solvated in dimethyl sulfoxide. Of course, it can work with chemicals in aqueous buffer. We have really good reproducibility across our 4-brain RosetteArrays. Our lumbar spinal RosetteArrays also show good reproducibility. And for some reason, they always tend to be a little bit higher than the 4-brain. Not sure why. but we have good Z-factor scores for those assays. We can integrate human metabolism or simulate human metabolism into chemical screens, for example, by doing a predigestion with the S9 pooled liver fraction. That's just a commercially available product. To date, our DNT screening with the 4-brain RosetteArray shows really good sensitivity and specificity, indicating its use for DNT hazard assessment. We've done now 2 different genetic models of spina bifida and 1 genetic model of autism spectrum disorder, and we are doing other precision medicine assays that we've been contracted to do from collaborations. So we think our RosetteArray platform is a really nice tool that could be addition to this DNT in vitro battery, particularly for detecting factors that occur very early in human neurodevelopment that can lead to both birth defects, neurodevelopmental disorders, and neurodegenerative disorders potentially. And that's where we have positioned this tool for its use. And I just want to say that you can access this tool if you're interested. Feel free to shoot me an email. You can either buy the plates and do this in your own lab. You can use our software that is available via the internet. Or if you're a company and just want this data for your compound, then we can provide that screening as well. We also do personalized disease model development. We have proof now for spina bifida and autism spectrum disorder, and we're doing this for other diseases as well for various research labs. And with that, I'd like to thank all the funding sources, including our funding from NIHS, which really allowed us to translate this via Neurosetta. All the people that contributed to this as collaborators, as well as former and current students. And with that, I'll take any questions.
Great, thanks. Thanks very much, Dr. Ashton. I had a couple clarifying questions for you, but we do have some questions in the chat or in the Q&A as well, so we'll let those gather and get to them. One of them was, I saw on a slide that you had this, it was about the ToxCast testing, and it said OECD 377 overlap. What does that refer to?
Yes, all the chemicals from, sorry for giving anybody seizure right now. All of it, the way it's presented, I can't just easily scroll.
Yeah.
All these chemicals are in that OECD guideline. So we made sure that we would be able to compare in the final publication to the detection from various assays in the OECD battery, the DNT IBB battery.
Oh, okay. Yeah, the guidance document. Yeah.
Yes. So this comes from the chemicals, essentially. EPA provided us a ToxCast library. We chose the ones that overlap with the DNT IBB guideline recommendation. Yeah.
Okay. And then you mentioned that you're using human S9. I've heard from some folks that it's difficult to obtain human S9, but you don't have any?
We haven't had any issues. We just get it from TempO.
Okay.
I mean, it might, I'm sure as with any product, it could be back ordered, but it's just a product we buy from TempO.
More expensive. Yeah. Okay. Cool. All right. So let's move on to questions. So first of them is, does the Rosette organoid contain non-neural cells like microglia?
From our analysis, no. So we are now growing these out longer. We don't see any microglia comes from a different germ layer. And so we see very, pretty much 95% or greater PAX6 SOX2 positive cells within these rosettes.
Okay, thank you. And stay on this slide because we've got a couple of questions related to this, I believe. So Anna is wondering, what is the reference based on for the screening performance? I think that
so we were looking at the DNT IBB as a major reference. Also, we did literature searches to see if there were results out there. This is not final, so I just want to put that out there. Essentially, we'll have to come to an agreement with our collaborator at the EPA as to what we call positive and negative, but this is our best guess currently.
Okay. And do you know, is that based on, I hear this coming from the OECD guidance, but is that based on human or animal, or you're not sure?
Yeah. So again, I'm not a toxicologist, but what I have learned from our EPA collaboration is it's based on both animal, largely animal studies, but some from the OECD guideline studies themselves.
Okay.
So those would be both human and animal cells.
I think they'd probably be animals.
So if you go back to the, so these NeuroCrest, these first 5 cell assays, along with this, these are all human. And the MPC 1 through 5 assays, those are those initially for the studies were human fetal derived. I think they've gone back now and done those with human pluripotent stem cells, or are doing those. The only ones that I know of that are, at least in the publication from my reading, that are animal are the rat cells indicated here.
Okay. Okay. I think what we're trying to, I think what we're like talking through, and we don't know unless someone is on, there may be someone on the call who knows. But is it like these in vitro results, or sort of consensus in vivo results? And I see Megan.
I think it's a mix.
I
So,
see
but
your
other
hand,
people might know.
Megan. Let me see if I can unmute you, if you would like to weigh in.
Oh, hi, Megan.
We can hear you. Yep.
Okay. So the list that Randy was working off of, that original list, it did distinguish between human data and animal data for developmental neurotoxicity.
Okay.
OECD will be releasing a new list very shortly that uses a more, let's say, thoughtful framework in order to say which chemicals you should consider first in regard to human relevant DNT. So that's a work in progress. But yes, the original list did include in vivo animal data as well as human data.
Okay, cool.
Yep.
Thank you. And while you're here, do you want to go ahead and ask your question? Because I was teeing your question up next anyway.
So I asked about the concentration response modeling, and you may or may not know this, Randy, but whether the data were fit bidirectionally. So I know for a lot of your data, you consider inhibition, which would be a negative response, but do you ever consider whether there could be a positive response and how that impacts your effect?
That is a great question, Megan. And one that I would have to say that I don't know the exact answer to that. I believe it was, so from the discussions that we had, I believe it was metric dependent, right? So for example, our control for single rosette emergence is typically 80% or higher, right? And so we typically for that one only do unidirectional because it doesn't make sense to go higher necessarily. But for the number of cells that we count per tissue image that we're analyzing, that could go higher. And so I think that was bidirectional. And so I think it was metric dependent for what made sense in the assay.
Okay, thank you.
Mm-hmm.
Thanks, Megan.
Yep.
Okay, next question is, are you applying artificial intelligence only for the image recognition within the Rosette platform or for some other predictive modeling like toxicity?
Yeah, right now it is for the images. We would love to collaborate with people to do the sort of more grandiose version of chemical space and stuff like that.
Okay, well, back to your may be inspired to collaborate with you. Okay, and you mentioned regulatory acceptance. Do you have any thoughts of what that looks like for this assay? I guess for specific, uses, like maybe your EPA data.
Yeah, so what we like to do is to we focus on the 4-brain RosetteArray assay. So when you're a startup, you have to focus on what you have money to do. So we focus on the forebrain to really try to push that forward. And so the chemical screen right now is for the forebrain, no metabolism, just run that assay. And what we like to go then further is to talk to ICVAM once we have that finalized about trying to do interlaboratory testing, and then go further, if that comes out well, to a draft OECD guideline, right? So we're trying to push in that direction, but that just takes time. So that's currently what the plans look like, in my head, at least.
Great. I know that the work that you've been doing is really focused on human, but this questioner is wondering if you've compared human and animal cell source to look at any differences.
Yeah, so the micropattern format, the sort of dimensions of the micropattern are optimized for human. For mouse, you will have to go significantly smaller. And we just have never, there's just limited bandwidth. So we haven't gone back to do that. But that is something that we can definitely do in sort of a technological feasibility manner. We just haven't done that. The other thing for mouse is it just requires a re-optimization of the entire platform, because mouse development occurs, even in mouse embryonic stem cells, occurs much faster. So this would be like a 2 to 3 day assay potentially in a mouse embryonic stem cell line versus a human. But we're interested in primates. We actually are thinking about sheep next, because at Wisconsin, we now have this really nice sheep model of neurotube defects. And so we might do sheep before we do anything else.
Okay. I'm going to get one more quick question, and I'll let you answer any more that come in by writing. But the question is, when you call active hits, do you consider the concentration relevancy to human exposure? For example, let's say if there's an active hit, but the concentration is way higher than what would normally be you would expect for human exposure. Would that?
Yeah, that's a great question. And I was trying to use the term hazard versus risk as much as possible, specifically because of that. So what we're doing, and what we do at the compound is look at hazard, at the company, sorry, is look at hazard. So we can identify if it has a hit in our assay, but the risk assessment comes in with knowing the exposure levels in the human population, right? And so that is something that we don't currently do, and we'll rely upon others to do that.
Yeah, okay. All right, well, thank you so much for your presentation. Thanks everyone for your engagement. And let me now introduce our next speaker, Laura. Dr. Laura Word was most recently, and Laura, go ahead and pull up your slides as I talk, and was most recently lead toxicologist at ICF International, where she applies her expertise in toxicology to inform risk assessments that protect human health and the environment. She previously completed her postdoctoral research at the US EPA, where she advanced new approach methods by using human high-throughput transcriptomic data to prioritize chemicals for safety testing. She has a master's in toxicology and a PhD in environmental sciences and engineering from the University of North Carolina, Chapel Hill, and has extensive experience in genetics, epigenetics, transcriptomics, and exposure science. Through her work in computational toxicology, Dr. Word strives to continue advancing innovative methodologies to enhance environmental health and safety decision-making. She is also looking for work, so please feel free to connect with her if you have opportunities. Laura, the floor is yours.
All right, thank you so much for that introduction. I'm really excited to be here and tell you all more about this research that I did. And the manuscript just became available last Friday, actually. So if anybody would be interested in learning more or going through the supplemental materials, I have the citation there for you. So transcriptomics data is really neat to be able to work with because we can use this to identify patterns of effect when we're exposing cells to different chemicals. And when chemicals impact the same target, the same gene, a lot of times we see similar gene expression differences that result because of that. And so we can use transcriptomics to help us predict bioactivity of chemicals without needing to use animal exposures, which is very, very powerful and very useful both within the field of toxicology and also in pharmacology if you're needing to do drug development and things of that nature. So a really brief overview of this from DNA that gets transcribed into RNA. We have messenger mRNA that's then translated into protein. And for transcriptomics, we're focused here on the mRNA levels. And I've conducted a comparison looking at RNA-seq versus TempO-seq technology for quantifying mRNA. With RNA-seq, this is the current gold established method. It is a non-targeted method, which is really great if you don't know exactly what you're looking for and you want to be able to capture novel transcripts, then RNA-seq is the way to go. But it has some really important drawbacks, including requiring purification of the genetic material before you can quantify it. And the RNA gets fragmented for sequencing, and that has to be aligned later on for data analysis, which is a very complicated step that requires really significant computing resources and can vary lab to lab as well. It requires very high level expertise. TempO-seq, on the other hand, it's much easier for the sample preparation because you can just use lysed cells. Less genetic sample material is needed in the first place as well. So if you have a very, very small sample, this is better. And you can even customize which gene transcripts you're actually quantifying because it uses detector oligos. And that is a positive there. If you are customizing, it can also be a drawback because if you don't have a detector oligo probe, then it won't quantify. So you're not going to get novel transcripts. But this is really scalable and can be less expensive than RNA-seq at very high scale. So previous research has shown TempO-seq to be very sensitive at detecting changes in gene expression, just like RNA-seq. And it also has very high reproducibility levels. Oftentimes it even had better signal to noise ratio and less unexplained variants, and has been consistent within fresh cell and tissue samples and also frozen and formalin fixed paraffin embedded samples. What all these previous studies have done though is using purified RNA, excuse me, for both the TempO-seq and the RNA-seq platforms. Nobody had compared lysed cells with TempO-seq yet to RNA-seq. And that's the gap that we strove to fill here. And also with using the full transcriptome and baseline gene expression for human samples and across a much greater spread of cell types than before. The TempO-seq data that I had access to was EPA's internal data. This was a phase 1 and phase 2 data set generated in collaboration with Unilever and Tox21, respectively. So again, this is baseline gene expression, and these two data sets were run at different read depths. So the phase 1 data was 6 million reads, phase 2 was 4.5 million reads, and it was generated a few months apart by the same laboratory technician, Clinton Willis, and were independent cell cultures, but were from the same cryostocks. The RNA-seq data that we used was Human Protein Atlas, HPA, which is publicly available, and this had about 20 million read depth. Notice that RNA-seq requires much, much greater read depth, largely because of everything being fragmented. So another thing to keep in mind when comparing these two technologies. And the very first step we needed to do was look at the TempO-seq phase 1 and phase 2 data and see if we could actually combine this for later comparing to RNA-seq. There were 6 cell types that overlapped between both of these phases of data. And the tissue origins are breast tissue, bone, liver, lymphoblast, colon, and lung tissue. For the person correlations, this showed very strong reproducibility. This was really great to see. I'm hoping you can see my my cursor here. So in the top left, this orange box, these are all the phase 1 technical replicates. It's technical replicate 1, 2, and 3 going down the diagonal, and then comparing against each other. So this first line here would be technical replicate 1 versus 2, and then on the top right is showing the Pearson correlation for that relationship. The bottom right orange square, that's showing the phase 2 technical replicates. And then in the blue is phase 1 versus phase 2 for that cell type. Panel A is just to help walk through everything. In panel B, this is showing those correlations across all 6 cell types that overlap between these 2 data sets. And what we found is the technical replicates had an average Pearson correlation of 0.98. And then when we're comparing across the 2 different phases that were at different read depths and the samples collected about half a year apart, the Pearson correlations were still 0.93, so extremely high reproducibility. Next, I ran principal component analysis as a dimension reduction method to help us visualize patterns within this data. And for TempO-seq phase 1 versus phase 2, this is grouping by the cell line and not by the data set. And then I ran some more tests to give us a sanity check and delve into this a little bit more. So panel B is a histogram of the principal component 1 rotation values. And everything outside of those lines at negative 0.02 and 0.02 are the genes that are driving the primary variants that we're seeing within PC1. So in panel C, I put those on a heat map to visualize. And what we see here is exactly what the PCA is telling us, which is that it's differences in gene expression pattern between the cell types themselves that is driving this variation that we're seeing. Because for each of these main columns in the dendrogram, it actually has 6 columns total within them for the 3 technical replicates across phase 1 in light blue and phase 2 in the darker blue. But these 6 columns are so consistent across those technical replicates and the 2 different data sets that they just kind of blur and look mostly like one set, which is really good. So this gave us really great confidence in being able to combine these 2 TempO-seq data sets to then be able to compare to RNA-seq, which is our step 2. So now we have 39 cell lines that we're able to compare to RNA-seq. I have in the second column here, the TempO-seq data phase. If it was in both data sets, then we just took the average across all of the 6 runs. But these cell lines now span 11 tissue types. Previously, there were just 4 tissue types covered, and the comparisons done between TempO-seq and RNA-seq. It was blood, breast, liver, and prostate cancer. But now we have even more tissue types represented than that. Plus, we have not just cancer cells, but also some hTERT immortalized normal cells as well. First thing I wanted to do is just get some eyes on the distribution of this data. So I plotted up some histograms And the overall shape of the data we're seeing between TempO-seq and RNA-seq was consistent, which is good to see. So this is showing for MCF7 on the top and U2OS at the bottom, with TempO-seq data being on the left, RNA-seq on the right. And when subtracting the data for each of the 19,290 overlapping genes for the TempO-seq log2 counts per million data minus RNA-seq log2 transcripts per million data, that is centered really nicely around 0, which is exactly what we would want to see if these platforms were performing similarly. Sometimes you'll see I'll refer to counts per million and transcripts per million jointly as expression per million, or EPM, just for a bit of ease when talking about these two, because it ended up being interchangeable. The CPM and TPM are comparable in this situation. So next up was to run principal component analysis for this comparison, the TempO-seq versus RNA-seq. And here we did see a very clear platform divergence across PC1. And I further ran PermaNova, which is permutated ANOVA statistical tests. I ran it with 999 permutations, and this was looking across all of the principal components for this TempO-seq versus RNA-seq comparison. And we saw that the platform effect accounted for 31% of the total variance, which is substantial. So really needed to delve into this more and understand what's going on here and which genes are they that are non-concordant and driving this platform divergence. The approach I took to answer that question was to stepwise and progressively remove those outlier genes that were the most different between the two platforms until the PermaNova variance explained for the R-squared, looking at platform effect across all PCs, was less than 10%. And that ended up being the 87th percentile and 13th percentile. So that stepwise progression, it would have started out a bit further, maybe removing genes outside of the 95th and 5th, then 90th and 10th, etc., until we got to this here, which met our requirement that once genes in the red here, that's outside of the 87th and 13th average percentile for the data distributions across all 39 cell types. Once that made the percent variance less than 10%, explained by the platform divergence, we knew we were good. So that came out to 3,810 genes out of the 19,290 genes total. And here's a heat map of what those genes look like, because it's entirely possible that a good number of these could be showing differences just because it's generated by different labs. But what we're looking at here is that across all 39 cell types in RNA-seq on the top and across all 39 cell types in TempO-seq on the bottom, is there's really strong consistency in either all of the cell lines having lower expression compared to the other platform or vice versa. That says to me it's probably a little bit more likely to be an actual technological platform difference rather than just because these cell lines were worked with in different labs. So at the time, scientific analyses involving these genes, if you're just working with the regular log2 data, then that should be interpreted with caution for now. And we have a list of these genes in the supplemental file available for everybody to download. We need other researchers to look into this as well and see if there's overlap in non-concordant genes found by other researchers too or not. Hopefully better understand what's going on here. And looking at the patterns of, well, are there any similarities in what these genes do that are non-concordant? I ran a gene ontology analysis and found that the odds ratios were really helpful here. So I had a minimum expression requirement. I wanted to make sure that with the gene ontology, we were looking at just genes that were actually expressed. And that was 10,487 genes that were at least 5 or more CPM in TempO-seq or 5 or greater TPM in RNA-seq. And for the gene signatures, we wanted that gene ontology signature to have at least 10 genes within it, and at least half of the genes within the signature needed to be within our list of expressed genes. And I'm going to walk through in a moment to show you visually what that looks like. So this resulted in 3,935 gene ontology signatures being retained in our analysis. This was from MSigDB, and the full list of 10,461 gene ontology signatures is in the supplemental materials. So then the odds ratios I calculated were the odds of one of those Go signatures being enriched with more non-concordant genes. Here's what it looks like for determining whether a Go signature stayed in our analysis or not. So this first one, you see that it has a total of 6 genes within the signature. That's less than 10, that doesn't meet our cut. The second one, it does have more than 10 genes within the signature. 8 of those are in our list, but 9 are not. So is 47% of the genes are in our list, and that doesn't meet the minimum cutoff of 50%. So that wouldn't be included. This next one is only 6 genes and none of them are in our list. So that definitely doesn't get included in our odds ratio calculations for anything meaningful it can tell us. And then the bottom 2 here, both of them have more than 10 genes in the signature, and at least 50% are within our list. In this case, all 11 were expressed genes that we have overlapping between TempO-seq and RNA-seq, and this had 13 out of the 16 or 81%. And here's the equation for the odds ratio calculation itself. It's looking at that ratio of non-concordant genes within versus outside of the Go signature, and divided by the concordant genes ratio within versus outside of the Go signature. So anything with an odds ratio greater than 1 was enriched for more non-concordant genes. And here are those signatures here. And this is with a false discovery rate corrected p-value that was significant. And a lot of these had to do with chromatin and ribosomes. Then the gene ontologies that were the most concordant had an odds ratio less than 1. And if we take the inverse of that odds ratio, that helps us with interpreting just how strong the relationship that was. These ontologies related primarily to cell structure, which is really encouraging. We want that to be consistent across cell types and technologies. So looking a bit more into those non-concordant genes, the signatures that consistently showed as non-concordant heavily featured histone genes and ribosomal genes. So for the histone, 73% of all of those genes were non-concordant. And this we can actually explain based on the technology. Because histone genes do not have poly-A tails, and RNA-seq preparation step normally includes a poly-A tail pull down. And so it makes a lot of sense why we saw low TPM values for RNA-seq. TempO-seq does not have that step, doesn't require or rely on poly-A tail pull down, and so it did a much better job at capturing histone gene expression. So if histones are important for the research that you're doing, then TempO-seq is preferable unless you do a lot of extra fancy steps with RNA-seq to not have that poly A tail pull down step. For ribosomal genes, more than half of those were non-concordant, but we don't actually know why. It's really unclear what's happening with those genes. It's possible that the TempO-seq probe design for a subset of those ribosomal protein mRNA genes just aren't reliably being captured. This is different from the rRNA. This is the actual mRNA for ribosomal proteins. But in this situation, RNA-seq had the much higher expression levels captured, and so it may be the preferable technology if you're looking at those ribosomal proteins. Now that we understand the platform divergence a lot better and what's driving it, the next question is if it's possible to resolve this platform divergence that we've seen. And for that, I calculated relative log expression. This looks at the log2 of the expression of that gene within a single cell line divided by the average expression for that gene across all 39 cell lines. And because of how division within a log works, that means we take those numbers and subtract the log values. So if the sample is 8 log2 CPM and the average across all 39 cell lines was 5 for that, then the RLE is 3, showing that the cell line you're looking at in that case had higher expression than the overall average. This did a beautiful job at smoothing out the platform differences that we saw, and then highlighting the individual expression patterns between different cell types. So on the left, this was before our RLE. You see everything grouped by platform, shown with TempO-seq being triangles and RNA-seq are circles. But after RLE, the data is grouping together by color, which is representing the cell lines. I just circled a subset of 5 here. So these are particularly cool. It is an offshoot that captures important biology because all 5 of these cell lines are cancer cell lines derived from the immune system. That was really neat to see RLE be able to really highlight that. And here are the Pearson correlations for TempO-seq versus RNA-seq. On the left in panel A, the top left block, those are TempO-seq versus TempO-seq cell lines for all 39 going across. The bottom right is RNA-seq versus RNA-seq, and the bottom left and top right are actually the same. That's showing the TempO-seq versus RNA-seq. And the diagonal in that bottom left square is really important to pay attention to because those are the Pearson correlations for the matching cell type across the 2 different technologies. So, for example, MCF7 and TempO-seq versus MCF7 in RNA-seq. And those initially had Pearson correlations of 0.77, which is strong and good to see. But the non-matching cell lines also had high correlations overall. It was about 0.64. And you probably expect that more or less. But after doing the relative log expression normalization in panel B here, we see that now is done remarkably well at really honing in on that signal for the differences between cell types. So now looking at non-matching cell lines, the Pearson correlation is almost 0, but between the matching cell type, I can again think of our example of MCF7 between TempO-seq and RNA-seq. That's 0.71, so almost the same as before. Still very strong, especially considering this is done in different labs. But the non-cell lines goes almost away for how much they match. And that's really powerful to use, especially after doing exposure. So this is all baseline gene expression data, of course, but if this was chemical exposures, that would be very helpful. So in summary, to recap what all I've gone through here, it started with looking at TempO-seq versus TempO-seq for our two different data sets generated at different times at different read depths, and it was very highly reproducible. And then for TempO-seq versus RNA-seq, 80% of the genes were comparable and concordant for the log2 expression per million data. And we evaluated that using Firmanova. Then gene ontology told us more about those 20% of genes that were non-concordant, showing it was primarily from histone and ribosomal gene families. And then PCA showed us how TempO-seq versus RNA-seq had that PC1 platform divergence, but it was readily resolved using relative log expression normalization. And that was without having to remove any genes, which is really good. And the caveat there is then you can't compare gene expression within a single cell type. You have to be looking across samples for different genes. And we can talk about that more if somebody's interested. But the last thing was the Pearson correlation showing how RLE accentuates the inter and intra-platform differences in cell line gene expression patterns. So for the strengths here were the first study to be able to compare these cell lysates for the TempO-seq data to purified samples in the RNA-seq. And we really expanded on the variety of the types of tissues that were looked at and showed very high consistency across all of the 39 cell lines for how TempO-seq and RNA-seq is comparing. Some of the complications is how this was data from different cell stocks and was generated by different groups. It's not entirely possible to pick apart how much of this variation we're seeing is because of that. But it's also a bit of a strength, really, because it's showing the real world reproducibility in real time that these technologies are having with each other. And it's still very strong. Another thing to caveat is about 8,800 genes weren't expressed at baseline in any cell type in either platform. They had less than 5 expression per million average overall. And so having toxicology studies that are inducing expression of more of these genes is going to be important for additional analyses. Of course, this has already been covered some by the prior research that's already been done, but those were always in purified RNA and not in the lysed cells. So that'd be a next step in research to expand on this a bit more. So in conclusion, found that TempO-seq was very highly reproducible at different read depths and showed consistent gene expression findings as RNA-seq. So hopefully this can help increase confidence in using TempO-seq data and also gives a framework for how to use RLE normalization to be able to actually even combine TempO-seq and RNA-seq data. For example, if we need to do a meta-analysis or want to compare results across different labs more readily. And then what we talked about here for future work needs to be done using the same cell stocks as well and inducing gene expression and see how the non-concordant gene list compares to what we found and try to understand what those differences might be in the technologies. So overall, thank you very much to all of my co-authors and the high-throughput transcriptomics team at EPA for their input on these methods. I've included a hyperlink to the manuscript summary and also the citation here if anybody downloads the slides afterwards. So with that, here's the poster as I presented it at ASCCT, and I'm happy to accept any questions.
Thanks, Dr. Ward. That was excellent. There's a group of us right now that are working on sort of developing IATA for systemic toxicity. And this is exactly the kind of work that we need to figure out, you know, when you vary your toolbox, what variations matter and which variations don't matter, and you know, how can we be flexible in some places and not flexible, you know, in other places. And so I think I really appreciate you know, the work that you're doing here. Go ahead.
Oh, that's really good. I'm glad. Yes.
I have a sort of a super basic question that you probably said, but I just missed, which is, does non-concordant mean a hit and a not a hit, essentially, or does it ever mean a difference in degree of expression?
Degree of expression.
Okay.
Yes. Yep. Since all of this is baseline gene expression data, nothing was categorized as hit or non-hit. We
Okay.
just had that basic threshold level of greater than 5 for being considered expressed or not.
Okay, great. All right, so the next question is wondering, based on what criteria did you choose the cell lines that you chose, the 18 cell lines?
That is a great question, and one that Joshua Harrell would be better able to answer. Those cell lines were selected prior to me joining the team. Who was it that asked that question?
Celia Voss, I think is how their name is pronounced.
Okay, I could follow up and
Okay.
share more information on it if they're interested.
Okay, thank you.
Mhm.
Next question is, the reproducibility of gene expression at mRNA level can be impacted by genomic DNA contamination, pseudogenes, and gene family members. TempO-seq performed using crude lysate may be more vulnerable to such reproducibility. Do you have any comment on those thoughts?
That is a bit out of scope for this particular project. I would be hesitant to comment on that particularly.
No worries. Okay, thank you. For relative log expression, would the tightening of the data be expected in varying tissue types of the same model or organism? I'm going to say organism. So if
Can you
you
repeat
have
the-
cells,
Mhm.
it would, for the RLE, would the tightening of the data be expected? Would you see the same tightening of that data in varying tissue types of the same organism, essentially? Like if you had.
Oh, I see. I see. Yes. If you have the same tissue type, but you're doing different chemical exposures, my suspicion is that RLE would still really help accentuate that signal of the different effects that the various chemicals are having on the samples, even within the same tissue. It might not be quite as pronounced as what we saw here across very different cell lines, but remember, even within these 39, there's good overlap with some of these being from the same type of tissue, and it's still very pronounced tightening after RLE.
I think they're also wondering about different tissue types. Like cell types from different, across cell types from different tissues, maybe for the same chemical.
Same chemical across different cell types. I have to think on that one a little bit more. That'd be interesting to learn more about what the research question would be and how to interpret.
I think it may, like for me, I think maybe it's stemming from this idea of, well, if you're trying to capture biology of an organism, let's say for systemic toxicity, how many cell types do you need to represent, you know? Maybe that's sort of where they're going with that, but
yeah, interesting question. I'd love to see more data on it. And
Yeah.
if anybody had data I could work with on it, I'd be happy to work with.
Yeah. So one last question. Would you anticipate similar results if compared to other platforms like DrugSeq or ScreenSeq? I don't know if you have experience with those.
I don't currently have experience with those other platforms, and would have to get into the actual data analysis to see if it does or not.
Okay, great. Well, thank you again for your presentation. Thank you all for your engagement and Q&A and discussion. Thanks both to Dr. Ward and Dr. Ashton for presenting today. Thanks to Clive for joining us and Allison for all your webinar support. Hope you all have a good weekend. And as we said, the PDF of the slides and the recordings will be up on the website, not today, but probably in a couple of days. So thanks, everyone. Take care.
Thank you.
Thank you.